So today our topic is compound microscope. So first of all, do you know what is a microscope? Does everyone know? Bolo kuch. You tell. What do you mean by microscope? Bolo, what is a microscope? ये definition नहीं पूछ रहा हूँ. What do you understand by microscope? Yeah, and don't sit like that. So a microscope is a device by which we can see the objects normally which you cannot see with your naked eyes. So what do you mean by a simple microscope? Because today we are studying the compound microscope, isn't it? So what is a simple microscope? Can anyone answer? Anyone? Anyone? Simple microscope क्या है? A microscope which is having only one lens. जो magnifying glass होता है ना, that magnifying glass is also a type of microscope. That is a simple microscope because there is only one lens involved. But this microscope, you can see that these are objective lenses. I will come to that part later. So these are objective lenses. These are some lenses, and above that there is another lens called the eyepiece, through which we see the object. So because there is a multiple system of lenses, one lens here, then after some space there is another lens. So there are multiple lenses. That's why it is called a compound microscope, and that's why it is not a simple microscope. So why we are doing this class? Why we are doing the microscope? Because in physiology practical, this is the very basic thing you have to know, and this is the very important instrument which you need to perform all the physiological activities or the experiments. Everything you have to do, any practical we are doing, after that we have to observe it under the microscope to see the results. That's why it is very important first to know the microscope fully and to make the proper utilization of the microscope. So micro means small and scope means to see. And that's why it is called microscope. So these are all these things are theory. You just can see it, what is written. No need to write all this, just look at it. It is everywhere in your book, in your practical books. So I have already told you it is a compound microscope because you have seen there are three lenses. There are three lenses pointing like this, pointing like little arms. So these are called the objective lenses. And there is a rotating nosepiece. These are the objective lenses. So there are few types of objective lenses. One is 5x. This is the slowest power. What do you do with the 5x? With the 5x, we set the field. Like suppose there is something called smear. Smear means you are taking out a blood sample and you are making a thin film to be observed under the microscope. It is called smear. So whenever you are making such smear on anything like that, staining means when if anything you are taking in your slide, to be able to see it under the microscope, that will be transparent. But if it is transparent, how will you differentiate that what is this, what is not? So we give a layer of color called stain, by which we can differentiate. Like different objects will take different types of stains. By that we can differentiate that this is the object, that is the object, that is called staining. So by taking a blood sample or whatever, like I am giving an example of blood sample only. By taking a blood sample, we will stain it, then we will put it under the microscope. So after that, with the 5x, what we will do? With the 5x, we will see that whether the position is correct or not, whether the cells or whatever it is, they are properly seen or not. It is in the viewable adjustment or not. After that, we will go to the 10x. In 10x, we can see the object a little bit more clearer. After that, there is 45x in these microscopes, and lastly, there is 100x. So these are the four compound lenses. Four objective lenses. One is 5x, one is 10x, one is 45x, one is 100x. But here, the magnification is little bit different. Like whenever you are viewing something with the 5x, the magnification is not actually 5x. It is 50x. Likely 10x. It is 100x, means the last result what we are seeing, that is 100x. Then with 40x it will be 400x, and likely in the 100x, we will be seeing it in a magnification of 1000x. Can you tell me why is it so? Because that in 5x we are seeing 50x, and in 10x we are seeing 100x. Why is it so? Is there any reason? Which magnifying lens? And yes, there is also called something called the eyepiece. I told you, it is a compound lens, compound microscope. So there is the part of the microscope called the body. Like suppose this is the body of the microscope, and these are the objective lenses. Above that, there is something called eyepiece, on which we keep our eye. That is called eyepiece. That thing also has a magnification of 10x. So obviously, when something is getting magnified with the objective lens, like suppose 5x, 10x, or 40x, that thing again is magnified in the 10x lens. So that thing comes as a multiplication of 10, like 50x, 100x, and so on. So this is the exact picture of a compound microscope. Everyone see the part. This one is the eyepiece, which I told that it will be of 10x power. Then this is the body tube. Body tube means the gap which I showed with this duster. The body will be in between the eyepiece and between the objective lenses. There will be this is called the arm. It is not written here. The conical C-shaped part, this is called the arm, by which we can hold the microscope. And the base is called the base, obviously, because the lowest part, which is touching the table, is called the base of the foot. Then there is something called coarse adjustment, fine adjustment. And revolving nosepiece, you got it till now? What is revolving nosepiece? Because there are 3 objective lenses, and we have to change it. Like suppose we are using the 5x. After that, I wish to use the 40x or the 100x or whatever. Then I have to change it. Then whenever I am changing it, I have to rotate the microscope. Like these lenses are there. By this one, I am seeing the object. After that, I have to rotate it. Then another lens will come. So this is the revolving nosepiece. This revolves and the lenses change. Then there is something called coarse adjustment, and there is something called fine adjustment. Then what is coarse adjustment? Coarse adjustment means whenever there is a big change you have to do in the microscope, in handling the microscope. Like suppose the object or the slide is very far away from the lens. So we have to pull it up. So in that case, we will use the coarse adjustment. That adjustment will be visible to our eyes. We can see that the microscope is moving, microscope part is moving. Then there is something called fine adjustment. Whenever after coarse adjustment, we can partly see the object. That the object is not that much visible, that much clear, but we can see it. So to make it clear, we will use the fine adjustment. Is it okay for everyone? For like a rough adjustment, we will use the coarse adjustment. After that, whenever we can slightly see the object, but it is not that much clear, to make it clear, we will use the fine adjustment. Then this is the stage. Stage means mechanical stage, means on which you will keep the slide or whatever you are observing. Then this is the condenser. Condenser means the area by which light rays will pass. I think you learned about microscope a little bit in your biology classes, na? That how microscope works. In those classes, we used to learn that microscope usually works with a mirror. There would be a mirror. In our time, there used to be a mirror by which sun rays would come inside and they would reflect into the microscope, into the viewing lens, in the eyepiece. They will get reflected into our eyes. Then we can see the object. But nowadays, that mirror is gone. Now we use a light source, which you can switch on and switch off. Then there is no need of the sun. We can use it in the night also. So in that case today, we are using the light source in place of the mirror. Then there is inclination, joint and everything. You can see all this. This is the description which is given, which I told you till now. This is the base. Base means the foot of the microscope, like this one, over which I am giving the microscope. This is the base. This is the arm by the C-shaped or C-shaped, or maybe sometimes it may be square also, rectangular shaped also. That thing by which we can hold the microscope. Then there is stage. This is the stage on which we will be keeping the lens, or keeping the slide. Then there is coarse adjustment knob, the big knob by which you can do rough movements, greater movements. Then there is fine adjustment. moves the stage or body tube very slowly for sharp, precise focusing. That movement we cannot see with our naked eyes because it moves like a few millimeters in a second. So it is not visible. The movement is happening. So this is the final adjustment. I've already told you these parts. This one is eyepiece or the ocular lens, which is having a 10x magnification. Then this one is the body tube. Body tube means this one is the body tube. This part, white part, that is the body tube. Then revolving nosepiece. This is the revolving nosepiece. This one, which revolves on turning. Then there is objective lenses, like I told you, 5x, 10x. These lenses, one will be 5x, 10x, 45, 100, like that. Now comes the illumination parts, means the light source by which we will be seeing the object under the microscope. One is Illuminator, means the light which you can switch on and off. Then comes the condenser. The condenser is the part by which the light is entering into the microscope. And there is diaphragm or iris diaphragm. Diaphragm means there is a part which you can control to make the light enter. Like you can allow for more entry of light or less entry of light with this diaphragm or iris diaphragm. We can also do it by adjusting the light source also, but this is an important part. It is included in the condenser itself. Now this is an important part which is required for your adjustment. Whenever you are viewing the object, viewing anything under the microscope, you have to follow these principles. Otherwise, you will be having a hard time to focus anything. So these things, the basic things, you have to keep in your mind. So one is 5x. I told you it is a scanning lens. Means you have to see whether the slide is perfect, okay or not. Whether you have to change the slide, whether you have to change anything or not. Then here the condenser position will be lowest. Condenser I told you na, that bulb-like thing, that blackish thing, that is called the condenser. That thing has to be lowered down at the lowest point. Whenever you are viewing with the 5x lens, that should be at the lowest point. And iris diaphragm should be almost closed. What will happen when it is almost closed? What will happen? Light कितना जाएगा अंदर? ज़्यादा जाएगा कम जाएगा? आइरिस डायफ्राम अगर एकदम थोड़ा सा ओपन रहेगा तो लाइट अंदर जाएगा ठीक से या नहीं जाएगा? बोलो कोई तो कॉन्फिडेंटली बोलो ना, इतना क्यों डर रहे हो? हाँ, so light will be very less because we are seeing scanning you don't need that much light in this area, so the light will be very less. Then we will use primarily the coarse adjustment because it is very downwards, na? The stage is very downwards. So for that, to bring it up, we will use the coarse adjustment. And immersion medium will be air. So sometimes you can just use a cover slip, or sometimes you will not. We will just view it directly. Then comes 10x lens. There is the magnification, total magnification given along with the eyepiece. So this is the 10x. Condenser position will be slightly above the 5x. Like suppose 5x, for 5x we have brought the condenser down here. For 10x we'll just take it a little bit upper. Then iris diaphragm will be mostly closed. In that was almost closed, it will be little bit more open. Primarily used for the rough focus, coarse adjustment. This is used for sharp focus. Okay, again this is the medium is air. We don't use any fluid or anything like that. Same for 45. One thing, the higher the power of the objective lens, the iris diaphragm will go upper. Means the stage we have to lift it up, more and more up. And the iris diaphragm will be partially open. For 100x, this is the highest magnification. For this, the iris diaphragm will be fully open. We have to allow the maximum amount of light which we can use for the 100x lens. For this, we are using the immersion oil. You can see there is clearly a difference. For the others, we used air, means we used nothing, just plainly we used to see under the microscope. But here we are using the immersion oil because we want the slide to be as close to the microscope as possible. And the oil, this oil is the oil we are using here is cedarwood oil, which is having a refractive index of nearly around air. So it doesn't distort the image. and whenever you are using the oil means there is the film, there is the lens, and in between there is oil, means there is no gap. So there is less chances of distortion. That's why we are using the pseudo dual for the 100x. And we cannot use any slide, or we cannot directly hit the 100x objective lens into the slide, because that will disrupt the whatever film we are making. Na? Now some precautions we have to take care of whenever you are using the microscope. First thing, we cannot touch the glass. Never touch the glass, otherwise there will be fingerprints, and you can because you are new, you will never know that why you are not able to view the object or whatever you are trying to view. So we should never touch the glass, that can spread fungus and they can destroy the glass. Always use silk clean cloth for cleaning, or not anything else, not your sleeve or anything, not in your dress. There are specific clean cloths that you have to use. 200 ARG. Whenever you are carrying any microscope, suppose these microscopes are already placed on a stand, I don't think you have to carry them. But suppose you have to carry them for any reason. So you have to hold the microscope with 2 hands, not with 1 hand. Otherwise it may fall down. So how will you carry the microscope? By one arm, you will grab the arm. Means this is the arm of the microscope. By one arm, you have to grab this. And you have to support the base with another hand, like this. And with 2 hands, you have to carry the microscope. This is the best way. Otherwise, the microscope will fall down and there are chances of accidents. The coarse knob forbidden zone. The coarse knob has one job at high power. It is to break the slide. Means at high power, you will not use the coarse adjustment, because already we will be using it to bring the object closer and closer. Because first we will be using the 5x, then 10x, 45. So obviously by then the level of the stage will be already higher. So we don't need to use the coarse knob at that place. We will just use the fine adjustment to just make the image more clearer. And spectacles. If someone is using spectacles, that person has to use spectacles. Because obviously if you don't use spectacles, you cannot view it clearly. So these are the takeaways. Now we will go to the lab and you will have a hands-on experience with the microscope. You will see the different parts. Let's go.
This is the condenser. What is the function of the condenser? It is focus on light on the object you are going to observe under the microscope. So if somebody and when using 100x, there is minimum or no gap between the slide and the lens, because we have to use citadel. It actually creates a film. Then the oil, then the it actually has the reflection index of glass, so that there is minimal or no reflection. We will bring at its topmost position and will completely open the eye. These parts we have to remove Because these questions will be asked. At what position you have to keep. You have to demonstrate it. The external is going to come, you have to demonstrate it. Then we'll place the slide, or otherwise the slide is already this nearby. If you want to see the blood corpuscles and everything, it will be already focused. We'll be focusing start from 5x, 10x, 45x and 100x.
कितना ऊपर करना है